industry. The way I've organized this session is to have the panelists give a very short presentation about their thoughts on the pharmaceutical industry and synthetic biology. And since this is really supposed to be a panel discussion, you can interrupt any time. We have all prepared very short talks, but this is supposed to be a free-flowing discussion. And so you can stop at any time if you have questions or comments. By way of introduction, so I'm Jeff Wei. The other panelists are Chris Anderson, Jingdong Tian, and Sean Jones. My own background is that I work for EMD Serono, which is a big pharma. We make protein drugs primarily, including Erbitux, which is an EGF receptor binding antibody that is used in colon cancer treatment, and we also make fertility hormones, and we make a version of interferon beta for multiple sclerosis. This is a relatively advanced, sophisticated set of drugs for a big pharma. They're all proteins as opposed to small molecules, but still, compared to the potential for synthetic biology, this is a relatively simple, primitive set of drugs. What I would like to do, first of all, just to because I made a guess about the audience, which is that you might not be that sophisticated about the pharmaceutical industry. So just out of curiosity, how many people know the difference between a phase 1 and a phase 2 clinical trial? Okay, so I'll go through that part quickly. So I'm going to review the drug development process and talk a little bit about precedents for synthetic biological drugs. I'll talk really quickly about what are likely barriers to bringing such drugs to market, and then also make a specific scientific comment about the different chassis that are being discussed with respect to potential synthetic biological drugs, or really more broadly about what we're using as chassis in synthetic biology generally and whether those are compatible with drugs. So the drug development process, as I guess half of you know, involves research, which can take arbitrarily long, a preclinical development phase that is informally called phase 0, and then phase 1, phase 2, and phase 3 clinical trials. And so these formal aspects of the preclinical development and clinical trials involve the following. The first part involves formal animal testing that you need to do to get approval by the FDA to go into human trials. When you're in human trials, the phase 1 clinical trial is supposed to be focused just on safety. Usually, if you're a drug company, in order to have everything be cost effective, you want to get as much information out of the trials as soon as possible to know if your drug is going to work and if it's worth investing more money in. So you try to get efficacy information, but the FDA really wants to know if the drug is safe. And this is usually a small trial of 12 to 24 people. Phase 2 is formally considered to be a dose-finding trial, and then phase 3 is usually a placebo-controlled trial. You find the optimal dose in phase 2, and then you match that dose against a placebo in phase 3. So one point about this is that basically this is going to be true of synthetic biological drugs as well as everything else, as any other drugs. Nothing about the formal drug process is going to change. And one of the complaints about the pharmaceutical industry, by the pharmaceutical industry and by the rest of the world, is that the process takes a long time. That is actually not going to change when we have synthetic biological drugs. We may have drugs that work better, but they're not going to get approved any faster. It may even take longer just because of the complex nature of the drugs. Now, there are a lot of precedents for synthetic biological drugs, and I think when we think about, we're always congratulating ourselves about how revolutionary synthetic biology is, but with regard to the pharmaceutical industry, you have to remember that over the past years and centuries, people have tried a lot of things for pharmaceutical purposes, including live organisms such as leeches, or for example, maggots are even sometimes currently used for wound cleaning. Also, there are vaccines that are really a good model for synthetic biological drugs. All of us probably have gotten vaccines that are attenuated viruses or attenuated bacteria, and these are extremely poorly characterized by the standards of synthetic biology. We may have gotten a polio vaccine, for example, which is an attenuated virus. I don't think anybody knows what the mutation is in the attenuated polio virus at this point. Also, the original polio virus that was developed by Salk and Sabin in the 1950s had SV40 as a contaminant. So in the past, people have received extremely poorly characterized biological agents as drug treatments and lived to tell about it. And the FDA has not freaked out. The population has not freaked out. There really is a lot of precedent. There's a lot of pathways that people have beaten in terms of what we would call synthetic biology, basically using organisms to treat disease. Another example from the past, so the kinds of things that we might consider would be viruses, bacteria, and engineered human cells in terms of synthetic biological drugs. We might also put highly engineered proteins at the very simple end of that continuum, but I think those are actually not much different from a lot of what's going on now that's not considered synthetic biology. And the real distinction, the real jump is between living and non-living treatments. The living treatments obviously present their own unique problems. Blood transfusions and various types of bone marrow transplants where you're transplanting live cells from one person to another represent another good precedent for synthetic biological drugs. And that's something that the FDA is familiar with dealing with and is very comfortable with. I would say the main issue for that most people in the audience might not be thinking about right now if you're trying to develop a synthetic biological drug is that the complexity of the drugs that we would like to develop. We would like to develop drugs that are able to do things that chemicals can't do. That's the whole point of synthetic biology. But as soon as you do that, then what you have to do is deal with a lot of essentially very tedious things that scientists usually don't like to deal with in terms of getting the drug approved. You've got to be able to show that there is reproducibility when you make a given batch of your drug. If your drug is a virus, how do you know that one batch of the virus is the same as another? How do you know that it can be shipped to the other side of the world? Or what are the circumstances under which it can be stored and so forth? And also, if it works by 5 different mechanisms, every batch of the drug is going to have to be tested and quality controlled for the activity with respect to each one of those mechanisms that you say is important for your drug. Also, I can speak personally for the pharmaceutical industry when I say there will be institutional resistance to synthetic biological drugs. Yeah?
I thought you just said that the point of synthetic biology is to develop things that chemicals can't do.
Right.
Aren't you going to use synthetic biology to develop chemicals? Isn't the drug still going to be a chemical?
No, the drug could be a bacterium. You could, and so so let me give a specific example, and this is really what I'm talking about. There is a drug that is approved. Let's eliminate my feedback here. Sorry. There is one particular drug that I think is way ahead of its time called Onyx 15. That is an adenovirus that is mutated in, I believe, E1A and E1B, so that it doesn't grow on normal cells. The function of E1A and E1B is to inactivate proteins like P53. So the resulting virus can only grow on P53 mutant cells. So you can put it in a person. If the virus finds a tumor, it will start to grow on those tumor cells, but it won't grow on normal cells. And that is really a good example, I think, of a synthetic biological drug. That's approved. It's not used for that many kinds of cancer, and so it's not a big money maker. It's not something that has gotten a lot of attention. But that is, I think, a good example of a synthetic biological drug. It's a live organism. Another example of a quote, drug that was tried and just turned out not to work that well in humans is salmonella that has been attenuated. A company called Vion in Connecticut developed a form of salmonella that is mutated in an aromatic amino acid biosynthesis gene. It's somewhat attenuated because of that. And the whole point about, and the goal was to treat cancer. And the idea about cancer specifically is that normally bacteria don't live inside your body at all, except for the colon and the gastrointestinal tract. Bacteria, your body is sterile. You don't have viruses or bacteria in your heart, your lungs, anything like that. Now, if you inject bacteria into the bloodstream, basically the immune system clears them extremely quickly. However, if you have a tumor, that as part of the evolution of a tumor, it spits out huge amounts of immunosuppressive factors to protect itself from the immune system. And if bacteria get into a tumor, they can survive. And I think Chris Anderson is probably going to talk about this more, so I won't try to steal his thunder. But there was this other company that has already done a phase 1 clinical trial where they injected people with Salmonella typhimurium, which is a mouse pathogen. It's closely related to a human pathogen. And the FDA was comfortable with that. That's another good precedent-setting example of a synthetic biological drug. But one point about that example is that I think there are going to be limited disease applications. We're not going to have synthetic biological drug live organisms for headache or the common cold. We're going to have them for really serious or fatal diseases, things like cancer, things like autoimmune disease where one of your organs has, essentially like pancreatic beta cells, are just gone because of an autoimmune attack. So you have type 1 diabetes. You need to replace those cells. If we make artificial cells that replace those cells, that's a synthetic biological drug, but it's for a really serious disease. I think that, and as I said before, I think non-issues, things that we might imagine could be issues but really will be non-issues, are going to be regulatory intransigence. The FDA has proven that it will be actually quite flexible about fairly complicated treatments. There are a large number of gene therapy clinical trials going on at any moment for cancer treatment. And also public acceptance. The public is very accepting of the idea that if you've got a fatal disease, then the FDA, then you can do almost anything to treat it. You know, being treated with live organisms, recombinant organisms, is easily morally justified, and the, quote, risk of contaminating the environment or something like that is minor compared to saving somebody's life. Now, I do want to make a specific comment about the chassis that we all use, like E. coli or any bacteria. The fact of the matter is that your immune system is designed, it's hardwired. There's a thing called the innate immune system that is hardwired to respond to bacteria, all sorts of features of bacteria. There are certain features of bacteria that humans don't have, and these are things like CpG dinucleotides. Okay, whenever you have a CG dinucleotide, it's essentially always methylated in humans, but in bacteria it's just not. Also, bacteria have N-formal methionine at the beginning of their proteins, whereas humans just have methionine, and so there is a hardwired aspect of the innate immune system that recognizes N-formal methionine, and also a bunch of things about the cell wall that are unique to bacteria. If we engineer E. coli to do anything, or really any bacteria to do anything, and then try to give it to a person, all of these things are going to induce inflammation, which is something that you really don't want. And so, for example, I remember last year at the iGEM competition, someone proposed to make artificial blood, which consisted of E. coli expressing hemoglobin. And that was a fine concept for a bunch of undergrads, but realistically, you would never, ever, ever want to do that. You would have, on the order of 10 to the 15th bacteria in your blood instead of red blood cells, and it just really wouldn't work. And also, you could say, well, we could overcome this problem. We're such geniuses that we could figure out how to eliminate all these immunogenic features of bacteria, but that is something that would obviously run into ethical issues. As soon as you make a bacterium or a virus that is really non-immunogenic, then you've created a potential pathogen that you really don't want the world to see. And so I think you have to be very careful when you think about suppressing inflammatory aspects of these chassis. So where does that leave us? We have these various choices of types of therapies, and they all have their specific issues. Gene therapy has the delivery problem. How do you get a given gene to essentially every cell in a tissue that is diseased? Viruses have the problem that they're probably the best, I think, in terms of being able to, for synthetic biological purposes, viruses have the fewest number of problems. But one problem that they do have is that they can mutate and evolve fairly easily. Another problem is that for any given natural type of virus, humans will have pre-existing immunity a large fraction of the time. And this is actually a practical problem in adenovirus gene therapy trials. A lot of people have been exposed to adenoviruses just in the course of getting colds and so forth. And so they will just have antibodies to the adenovirus that's being used as a therapy, and it just doesn't really work. Bacteria, as I say, there is this problem of inflammation that may limit their use to cancer. I think the use of bacteria to treat cancer is extremely attractive, but that might be really the only use. And finally, cell therapy, such as replacing pancreatic beta cells, replacing cells of the substantia nigra for Parkinson's disease, those have, what you're not really going to be able to do is to manufacture a cell line that you can use, because also you have the problem of genetic variation from person to person. And so realistically, the synthetic biological therapy is probably going to consist of taking out a person's cells, doing something to cultured cells from that person, and essentially giving them an autologous transplant of engineered versions of their own cells. So those are some thoughts. Unless somebody has questions, I can hand off to our next panelist. Chris, you want to take it away? Oh, yeah? Yes.
Is it possible that just for a small period of time you artificially suppress the immune system, then treat the patient, and then,
what, with the bacterium?
Yeah, or like with adenovirus.
Okay, so the question is, would you want to combine immunosuppression with a treatment with one of these organisms? And I think that would be pretty risky. It would depend on the disease, but that seems like it would be pretty risky because usually when the immune system is suppressed in the context of a traditional therapy, it's usually the acquired immune system that is, for example, being suppressed when you have a transplant. And so you will suppress T cells. When you start suppressing the innate immune system, that is a much more profound thing that you're doing to the body, and you have your innate immune system always functioning because your skin and your intestine are just full of bacteria. And so if you compromise the innate immune system, I think that's really practically pretty dangerous.
I think in terms of the general architecture of the field as it applies to synthetic biology, or synthetic biology as it applies to therapeutics. I mean, clearly there are a finite number of substrates that we work with in synthetic biology. You're either working with viruses, those could be bacterial viruses or mammalian viruses. You're working with bacteria, yeast, or mammalian cells, and that's really all we have. So clearly it's the case that is the subset of things that are worth talking about. But if you're talking about bacteria specifically, it could mean several things as to what you're doing with the bacterium. And I think the one that, first part of my talk here is very similar to Jess. Clearly, traditionally, what it has meant, bacteria in the context of therapeutics, or engineered bacteria in the context of therapeutics, has been you create a gene that encodes some therapeutic protein, you put it into a bug, you express the protein, and you sell it for billions of dollars. But that's changing a little. First of all, we have this kind of second generation small molecule biosynthesis in microbes, like the artemisinin work by Jay Kiesling's lab, where again it's a purified product from a bacterium, but it's something that's fairly advanced engineering to create this organism, creating the product that then gets put into a human being. But the one that I'm particularly interested in is kind of the third scenario, where you have bacteria as live therapeutic agents. But there's kind of this intermediate scenario where you're using dead bacteria of some sort. So they don't have any viability whatsoever, but it's a complex product that's being injected into a human being. I think one thing I would disagree with Jeff on is the area of modifying the immune responses to the bacterium. I mean, if you have something that is bacteria-derived, either a ghost cell that you really could show there's no DNA left behind in these organisms, or a lot of people have worked on mini-cell type things derived from bacteria, which again have no DNA in them, then the risk of creating something that has fairly low immune responses in a human being is dramatically reduced. So I think that may in the future be a viable product, but just in terms of doing it, I can tell you from working in this space for a little while, it's going to be really hard to get there. But I wouldn't rule it out that 30 years from now, those are the sorts of things that when we meet, we're talking about. The one specifically we work on in my lab, and I've been doing this about 4 years now, is tumor-killing bacteria. So it's analogous to the work done by Vion, but it's a ground-up approach in E. coli rather than sort of a top-down and build-back-up approach that they do in Salmonella. But in general, these agents are supposed to be injectable into the bloodstream, localized to and invade cancer cells within solid tumors, and then kill them. And the general approach to this is to treat it as a synthetic biology parts device chassis type problem. So the real mainstay of the project is to first of all create some derivative of the lab strains of E. coli that has a decent survival within the bloodstream. I mean, it's, as you said, your bloodstream is a sterile environment. It's fairly difficult to make an animal a bacterial agent that you're going to put into an animal and have it survive for anything more than about 5 minutes. So that's actually a major hurdle in this work, is just getting it to last long enough that it gets to the tumor. And that really, I think, if you follow the literature on Vion and the human trials and the animal trials previous to it, where that stuff really fell down was around the issues of the differential clearance rates that you get between a human being and a mouse, and they just got wiped out when they put them into human beings for the most part. But in general, this idea of using therapeutics derived from bacteria, it seems like an odd idea. For some reason, it elicits a yuck factor that even talking about viruses as therapeutics doesn't elicit. And I don't totally understand that, but it's clearly the case that there's a long history of using bacteria as therapeutic agents. So if you go into your grocery store right now, maybe not in Hong Kong, but maybe back in the States. This was in North Carolina that I took pictures. So Activia is this yogurt product that you can pick up in your grocery store, and people intentionally infect themselves with bifidobacterium with an expectation that it will improve their digestion. And it's actually a popular product. So about a year ago, I went back to the grocery store, and they've expanded their line. You can get lots of different flavors. Now they have cheeses and yogurts. People are really buying this stuff, and it doesn't bother them one bit that there are live bacteria within these products. And on kind of the higher-end therapeutics and the sort of systemic therapeutics, well, this one isn't systemic, but it's a very serious sort of treatment. If you're ever to be so unfortunate as to get bladder cancer, the primary treatment for acute bladder cancer is a bacterium called TICE, or Mycobacterium bovis BCG, and they catheterize this material. It's a live bacterium. They catheterize it into your bladder, where it elicits a specific immune response that results in the destruction of the tumor. So now, if you're talking about injecting bacteria in the bloodstream, it's an example of a very smart drug. And you can think of these agents as being analogous to the smart liposome products that have been kind of popular within the more chemical side of these complex drug areas. So a lot of people work on these sorts of things that are, a lipid bilayer surrounding some really nasty toxic compound with antibodies decorating the surface that help localize them to the surface of cancer cells, where hopefully something happens and the small molecule is released locally, killing the cancer cells. You can think of bacteria as a very similar agent, where they're going to be passed around in your bloodstream. You can have binding specificity by putting things on the surface. You could have environmental sensors that respond to chemical cues. You can have dynamic responses in response to sensing events, such as the delivery of toxic proteins. They can do logic and communication, so they can process signals. And they have this very special property: bacteria can swim. That is something that you just don't get out of a chemical species. So there's a lot of particular intrinsic functionality that is possible within this object that is a bacterium that just doesn't exist within other things. So the sorts of applications, I think principally I agree with Jeff, like the primary application here is anti-tumor agents. But you can also imagine targeting bacteria towards single-cell diseases like HIV-infected cells, targeting the cells that are the bad leukocytes associated with autoimmune disease, leukemia. But you can also think of ex vivo strategies. So you talked about the possibility of ex vivo sort of applications where you pull cells out of a human being, do something in vitro, and pass them back. That do something could be infect them with a bacterium that has a very complex program that reprograms them to do whatever it is before you put them back. You can also have things that are sustained hormone delivery. So I recently saw a paper where it wasn't entirely a synthetic biology sort of paper, but it turns out there are things you can put in a bacterium and then have the person ingest them so that they're in your gastrointestinal tract. And the things that these bacteria are engineered to secrete will elicit the release of insulin within your gastrointestinal tract. So there are a variety of kind of bizarre applications like that, which possibly don't involve systemic infection, which I think from a safety standpoint is what kind of gets people, is the idea of putting these in the bloodstream. But if you're talking about putting on the surface of the skin or in the gastrointestinal tract or the lungs or the ears or something, it seems a little less scary. Also, you can have bacteria on bacteria agents potentially. For some sort of things that might make sense, tuberculosis or other. There are things in that same category of desperate life conditions in that area of bacterial infection that potentially using a bacterium to kill the other bacterium might make sense. And finally, although you disagree with the idea, 30 years from now, maybe blood substitutes. We'll see. I don't expect to see that anytime soon. That'd be hard. All right, finally, but you could have really kind of superficial therapeutic bacteria for things like hair loss, hair removal. If you're talking about agents that don't involve anything systemic, where there just isn't this sort of health scare issue, there's no reason to think that a biological agent would be a bad way to approach those problems. And finally, this is kind of the classical area, but probiotics. You could have bacteria that just make things like beta-galactosidase that people eat. And that's relatively harmless sorts of drugs. All right, so why bacteria? There are advantages to all these various types of things that you might put into your body to elicit disease. In my mind, the big thing that is special about bacteria that makes them desirable for some applications is the amount of memory that's available within a bacterium. So it's at least a megabase in E. coli, possibly significantly larger. If you think about that, that's about 1,000 genes you get to play with. That's a lot of room. I mean, that definitely, in terms of our abilities to build systems, there's far more space available in the organism than we can exploit today with our abilities to design. Also, there's a very deep literature based about how bacteria, natural bacteria, interact with human systems. So it's not like you're working in the dark, not knowing what's going to happen when you put a given set of genes into E. coli. There's a lot of literature that will give you some predictability as to what's going to happen. And it's inherently modular. So I feel like this is something that's very different about the way bacteria work from the way viruses work. When a bacterium does something to a mammalian cell, it'll have 5 different sets of genes, all doing a different thing. So for example, we'll have one set of genes that puts capsular polysaccharide on the surface and blocks the immune system from clearing it. Then it'll have a separate set of genes that are responsible for letting the bacteria adhere to the surface of an epithelial cell. Then another set of genes that's associated with getting them to invade and go inside. And there would be yet another set of genes if something happens once they get inside. It's very different from viruses where you tend to have single proteins that are playing very diverse roles within the virulence. But just fundamentally, they're dynamic sensor actuator packages. That's a distinct advantage over chemical systems, just that they can change. They're dynamic. And finally, it's easy to engineer bacteria to work in an environment like the bloodstream or inside the cytoplasm of a cell, because you don't actually have to have the mammalian cell there to see the phenotype of the bacterium. The bacterium's viability has nothing to do with the mammalian cell. It's totally orthogonal to it. So you can create these kind of surrogate environments in vitro and rapidly assay what the bacterium is going to do in that sort of chemical environment that's going to encounter. And from a safety perspective, one big advantage of bacteria over viruses for various applications is that you know the biotope of the bacteria because you made it. So you know what it's resistant to and what it's sensitive to. And you can usually kill them with antibiotics, which are highly orthogonal to all of mammalian biology. So I think inherently there's some safety benefits of using bacteria.
I'm going to briefly talk about some new technology development in synthetic biology and how to apply it to solving some of these problems, like develop new vaccines for emerging infectious disease. So I'm from Duke University, and this work is in collaboration with the Duke Human Vaccine Institute and CHARVI. CHARVI is a global consortium funded by NIH to solve this HIV/AIDS problem. So we are collaborating to try to use a new synthetic biology approach to see how we can do on the vaccines. So my lab mainly focus on developing the high throughput gene synthesis technology that you have heard about throughout the meeting. And so we develop those technologies and also develop new applications for those, and this is one of it. So why we are working on this vaccine development on emerging infectious diseases, so such as pandemic influenza, even flu, HIV/AIDS, SARS, etc. I mean, the bad things about this is currently there's no vaccine against it, right? Because I mean, this virus is mutated so fast, and the current vaccine development is the conventional technology is just too slow. So we're trying to look for solutions to that. I mean, this is just the thing from the past pandemic. I mean, throughout this century, there have been 3 major influenza pandemics. So one in 1918 and 57, 68 killed about 40 million people, 1 million. And there's a study published that predicts that if there's another pandemic happens today, the death toll would be about 62 million. So without a vaccine, I mean, that's unthinkable. So that's for pandemics, and just look at the common flu. I mean, each year, I mean, the flu season is coming, right? So each year, just in U.S., you have to make about 80 million doses annually, right? And those vaccines are live inactivated viruses, and it typically takes about 6 months to grow, right, in eggs. So, I mean, according to CDC, each flu season, there will be about up to 50 million people get sick, and 200,000 hospitalized, and 36,000 deaths. The cost, I mean, for treating that and preparing the vaccines would be about $10 billion per year, and so. And that's just common flu, right? And if a pandemic strikes, it could be much worse. So this is just a computer simulation simulating that if 10 person with the infection landed in, for example, Los Angeles, it would take only about 3 months to get the whole country infected, as shown in the red. So obviously, I mean, if you take 6 months to develop, just to grow up, not about developing a vaccine, just to grow up a vaccine, obviously that's too slow, right, for a 3 months development period. So that's why, I mean, people have an interest in developing those fast response vaccines. So we pretty much have covered this slide to compare the conventional vaccine with the fast response vaccines. Mainly, I mean, conventional vaccine is just too slow and costs too much to develop. And the fast response vaccines, mainly we're talking about the DNA vaccine and some related. I mean, they have the advantage of being react fast. So because, I mean, compared to either live vaccine or proteins-based vaccines, the DNA vaccine has the advantage of you can. I mean, it's DNA, right? You can just grow the bacteria up and isolate the plasmid. So that's very fast, and they're easy to store because it's DNA, right? You can store it without refrigerator, just put it in the tube. But there are technical challenges, right? The main challenge is the efficiency is very low, and you don't get very high efficiency, mainly because you don't get very high, I mean, expression level and things like that. So that's why, I mean, we think that our technology can help. So that's why we are using this so-called ultra high throughput gene synthesis and optimization technology, trying to solve this problem to be able to efficiently optimize the vaccine, the antigen, so to get very high efficiency. And so I'm not going to go in too much into the technology itself because of the topic that we're going to cover. So I'm just quickly go over the strategy that we're using. So what we do is we do high throughput gene design, meaning we can design a lot of versions of the gene, of the antigen, and then use the high throughput gene synthesis technology so we can synthesize them very cheaply and in a high throughput fashion, and then we do an expression screen or assay to quickly identify the best clone, right? Have the highest efficiency. So we already did the proof of concept experiment using on this technology in both E. coli and in human cell lines. So this is just showing one experiment in E. coli where we optimize one test gene to see to show that this technology works. So in the on the right is so this gene, if you express it in E. coli, it makes a blue color, right? So just by the color intensity, you can tell how much protein is expressed. So here we designed, synthesized this. Here shows about 1,400 genes, the same gene, but just different codon usage, right? So you can see a spectrum of expression level from the top is the highest, the bottom is the lowest. And if you monitor the protein expression kinetics, so this is 1,400 of them. I just just plotted about 8 here, showing you, you can get a spectrum of expression. So, and then you can just pick the top one, right? That's the highest efficient sequence that you want. It gives you the highest expression level. So there's no guess involved, like the current codon optimization, where you send out order to the company, they send back you a design of a sequence, then you synthesize it, you test it, and if it doesn't work, you have to go back and do that again. So this eliminated those repetitions. Just through one round of design and screen, you can get the best, the most efficient one. So we did the same demonstration in human cells, and again we showed that. So this is about 5,000 genes that encode the same protein, but with different codon usage. Here we can show that we can, using the facts, to demonstrate that we can isolate the most highest expressing clones. So with this technology, we are ready to apply it to vaccine design. So mainly we want to use that in DNA vaccine, but in this example that we did together in collaboration with the Charvey and Duke Human Vaccine Institute, is we're trying to optimize a new HIV vaccine that they are trying to develop. You heard about the bad stories about the failures about HIV vaccine, right? So this is a new version that they're trying to develop, where they want to express the one of the HIV antigen in the bacteria. It's the mycobacteria that you just mentioned, right? ECG, and because that bacteria not only induce the immune response from people, right? So if we express the HIV vaccine on the surface of that bacteria, that can it can use the bacteria as a vector to induce the human immune response. So, to quickly, the bad thing is, the HIV, unfortunately, the antigen does not express very well in this bacteria. So we have to quickly optimize that, and using the same technology that I talked about, we quickly, isolated the best expressing sequence and put it. We can demonstrate that we can get a high expression of this, and then so we isolated the clone that has the highest expression level of this protein, and the next we are testing. This is currently an animal test. We're testing whether they can in animal it can induce the immune response that we expected. So this just demonstrates the very early stage of drug development. You get a lead, you go through the animal test, and then go to further clinical trials. So I will stop here, and if you have any questions, the next.
You can interrupt with any questions for anybody. Usually when there's a panel discussion, there's actually a table where we all get to sit and face the audience. Yeah.
So, Dr. Tia, can you explain how this vaccine is going to work? Because the virus mutates quite rapidly. So even if you are expressing one particular antigen on some vaccine surface, and the immune response is specific to that.
Right.
And by the time your immune system responds, the virus may change.
Right. So we have developed this technology mainly for the DNA vaccine. In that case, you can quickly once the virus mutates, you can, now the new sequencing technology can allow you to sequence the new, virus sequence in a day, right? You find that out, develop the new DNA vaccine, and that's going to be very quick, right? If you have this kind of technology to optimize its expression. And so you're talking about this particular one, put it in the bacteria, and that's going to be a little bit slower, but first you have to find a way that you can. Right now we don't have any vaccine against HIV, right? So anything works, and we worry about mutation later.
You just mentioned that you can anchor the vaccine, displace it in the surface of the bacteria,
right.
And will that affect the bacteria's growth?
Not really. We didn't notice that. It shouldn't, actually.